Hello, my name is Abby Gilham. I go to Eastlake High School, and for my Student Bio Expo project, I study naturally occurring antibiotic resistance. So what I did in my lab research was that I characterized the bacteria found on human skin, inspired by research of the Microbiome Project. I did a much more basic version of what they're doing, and swabbed bacteria off the inside of my elbows and studied it through gram staining and a bacterial lawn with an antibiotic tablet. In this bacteria, I was looking for antibiotic resistance. My first step was to swab the bacteria off of my skin and let the colonies grow and observe them. These two plates were swabbed from the inside of each of my elbows, and as you can see, the bacteria grew in colonies. And if it's showing up right here, some of them are yellow and some of them are white. This became significant with labeling later on. The second step of this process was to transfer cells into liquid media, which involved choosing a colony and transferring it. So I'm going to skip back a slide really fast. As you can see, again, some yellow colonies, some white colonies. I picked a yellow colony and a white colony from each plate to transfer into the liquid media. The media incubated at 37 degrees for about 24 hours before I could begin the rest of my experiments. The third step in my lab research was gram staining, and this is the first step that involved data and observations. I was looking for the shape and color in this process. Gram-positive, which shows up as purple, or gram-negative, which shows up as pink, meaning also that gram-positive or purple bacteria is antibiotic resistant and gram-negative bacteria is not. The fourth step of my lab research was to make agar plates, which I had to do on my own for the first time, and to spread 100 microliters of the liquid cell cultures onto these agar plates. These were spread, and then an antibiotic tablet was added. Penicillin, ampicillin, and tetracycline for each of the 4 colonies of liquid media that I grew. So that's a total of 12 plates in all. And my final step was to make observations and measure my data. I observed the gram stains under a microscope and found a combination of gram-positive and gram-negative bacteria. I then observed the plates, the patterns in which the bacterial lawns grew, and specifically measured the empty space surrounding the antibiotic tablet where bacteria would not grow because of the proximity to the antibiotic. Hopefully that makes more sense on this slide. These are all 4 sets of plates that I had. These cells here are from a white colony off of the first plate, from a yellow colony off of a first plate, white colony off of the second plate, and yellow colony off of the second plate. So you can see going in right here, bacterial lawns grew on all 3 of these plates. The place where I measured was right across here, right across kind of the diameter of the circle, right here. Right there, ampicillin and penicillin, as you can see here, were about the same distance, and tetracycline was quite a bit bigger, so clearly there were fewer bacteria that were resistant to tetracycline. P1Y, or the yellow colony from this plate, these bacteria, interestingly, were the only ones that grew in colonies after being transferred from the liquid media. And you can see that these bacteria did not mind penicillin and ampicillin very much, although they did give tetracycline quite a wide berth. That's what this line is right here. This is all empty. And I don't know if it's showing up on here. I don't think it is. But inside of this penicillin circle, there's actually a few colonies that are very close to the antibiotics. So yeah, clearly antibiotics don't bother these cells very much. And then this one, similar patterns. Less resistance to penicillin than there is to ampicillin, though they are both less than tetracycline. And on these last set of plates, again, similar patterns, although the tetracycline tablet unfortunately fell off during incubation at some point. So the size of the little space right there is about the same as the penicillin and ampicillin. But again, we have no idea of knowing how long the tablet was on there when that happened. So, oh, sorry about that. Here are some of my gram staining results. I was not able to get pictures of the gram stains, unfortunately, so I drew them to the best of my ability. This was from the first plate with the white colonies right here. And this was the pattern. These kind of star circle looking things are the cocos shape, and these kind of lines are supposed to be the rod shape. So this one had quite a bit more gram-positive bacteria. Oh, and also black is supposed to, just pretend that that's purple because I did not have a purple pen when I was making these drawings. So black in these drawings is the gram negative, is the gram positive, excuse me. Second plate, the white colony had, let's see, that was quite a bit more of a mix of gram positive and gram negative, and this was primarily the cocos shape, although some rods were present. First plate with the yellow colony was primarily made of cocci, except for the rods right on the bottom here. And the second plate with the yellow colony was pretty much made up of all cocci shape. They kind of looked like dots in a smudge. That is a fairly accurate depiction, even though it was just with ballpoint pen. And excuse my little cheat sheet notes down there. I keep mixing up which stains are which and what they mean. But as you can see, there's a presence of gram-positive and gram-negative bacteria in all of these colonies, which really helps explain their resistance to some of the antibiotics and helps show that many, many different bacteria can exist, inside of a certain colony and on the human skin. So, conclusions that I drew from this lab. First of all, there's enough bacteria on my elbow to grow an entire agar plate, which is pretty cool. The bacteria grew in colonies on the plates, which I was not sure about, and that was interesting to see. And that some antibiotic resistance, as seen through the plates and through the gram stains, was present, and that these antibiotics were the most resistant to penicillin and the least resistant to tetracycline. One theory I have on that is that penicillin has been around longer than tetracycline has, and that perhaps these cells acquired their resistance through transformation or transduction with human cells, and more human cells are resistant to penicillin because of how long penicillin has been around. But future research studying specifically what makes these bacteria antibiotic resistant would be fascinating, as well as studying antibiotics with the skin cells directly. But anyways, more research for another day. I've talked enough on this one. Thank you for your time, and please feel free to read my research paper for any additional information.